This video explains how bacterial and yeast samples are prepared for MALDI-TOF mass spectrometry with the VITEK MS system. At the end of this video, you should be able to list the VITEK MS reagents and disposables needed for preparation of bacterial and yeast isolates, correctly perform sample preparation for bacterial and yeast isolates. Step 1. Assemble all materials. These include VITEK MSDS target slides. Target slides are stored inside the plastic cartridge at 15 to 25 degrees Celsius until the expiration date. Each slide contains 48 sample spots divided into 3 sections of 16 spots each. Each section is called an acquisition group. Each acquisition group has a small spot in the center of the group to be used for calibration. VITEK MS CHCA matrix. Each box contains 5 0.5 milliliter yellow-capped vials of matrix. Store the matrix at 2 to 8 degrees Celsius, protected from light. Matrix vials are good for 1 week after opened. VITEK MSFA formic acid. Each box contains 5 0.5 milliliter red-capped vials of formic acid. Store the formic acid at 2 to 8 degrees Celsius. VITEK MSFA vials are good for 2 weeks after opened. 1 microliter calibrated loops. Precision pipetters to deliver 0.5 and 1 microliter. Sterile, colorless pipette tips without filter. Microtube rack. Powder-free gloves. A good light source and/or magnifier is not required, but is helpful when preparing spots. E. coli ATCC 8739 calibrator organism. Reconstitute a purchased stock strain according to manufacturer's directions. Inoculate 2 trypticase soy agar with 5% sheep blood or 2 Columbia agar with 5% sheep blood plates and incubate for 24 hours at 37 degrees Celsius. Label the plates Mother Plate 1 and Mother Plate 2 and store at 2 to 8 degrees Celsius. On days 1 through 7, subculture from Mother Plate 1. Dispose of the mother plate properly after day 7. On days 8 through 14, subculture from Mother Plate 2. Dispose of the mother plate properly after day 14. A fresh 18 to 24-hour subculture of the calibration strain taken from the mother plate and grown at 35 degrees Celsius plus or minus 2 degrees Celsius on trypticase soy agar with 5% sheep blood or Columbia agar with 5% sheep blood is required each day of testing on the VITEK MS. The calibration strain is required for every acquisition group used for testing on every target slide. Additional bacterial quality control if required by your lab procedures. Enterobacter aerogenes ATCC 13048. Negative bacterial control. VITEK MS CHCA matrix alone with no organism. Additional yeast quality control if required by your lab procedures. Candida glabrata ATCC MYA 2950. Negative yeast control. VITEK MSFA plus VITEK MS CHCA with no organism. Test isolates, bacteria and yeast. Fresh 18 to 72 hour bacterial or 24 to 72 hour yeast culture on a validated media. Validated media include Columbia blood agar with 5% sheep blood, trypticase soy agar with 5% sheep blood, trypticase soy agar, chocolate polyvitex agar, campylosal agar, McConkey agar, modified saccharide dextrose agar, Chrome ID CPS. Note: VITEK MS testing of bacterial and yeast isolates cannot be performed directly from clinical specimens or with mixed cultures. Step 2. Adding the calibrator to the calibrator spot. Once all the materials are assembled, put on a pair of powder-free gloves and place the VITEK MSDS target slide on a flat surface. Be sure to handle the slide only by the edges. Avoid touching the spots on the slide. And do not write or mark on the slide. Using a 1 microliter loop, pick up part of a suitable colony of the fresh E. coli ATCC 8739 calibrator. Do not pick up agar along with the colony, as this may lead to calibration failure. Apply the calibrator strain to the center of the calibrator spot of the acquisition group that will have patient isolates applied. Depending on the amount of material selected, dabbing 2 to 3 times with the loop works well. Insufficient quantity of colony applied usually results in no spectra being acquired. If an excessive quantity of colony is applied, cells may not suspend well in the matrix suspension. Little or no crystal formation occurs with the matrix. The acquisition process may be hampered. Immediately, flip the loop and smear the calibrator in a thin, even layer on the spot. Be careful not to spread the organism outside the spot limits. Good smear quality is essential. The calibrator is the first and last spot tested on each acquisition group. The goal is to acquire 100 profiles. If less than 30 profiles are obtained, the calibrator fails. If the calibrator fails, no patients are tested in that acquisition group. Discard the used loop in an appropriate biohazard container. Open the VITEK MS-CHCA matrix file. It is not necessary to warm the matrix to room temperature. Immediately after spreading inoculum, add 1 microliter of clear matrix to the center of the spot. It is important that the sample is not allowed to dry before adding the matrix. If too dry, the sample does not stick to the spot surface and may peel off. The sample must be moist when the matrix is added to obtain the even distribution of sample and matrix required for crystallization. Yellow crystals present in the matrix do not affect the reagent performance, but only clear matrix solution should be added to spots. Avoid the yellow crust on the rim of the vial and avoid aspirating the yellow pellet at the bottom of the vial. The matrix contains a high concentration of organic solvents. Close the vial after dispensing the reagent to avoid evaporation. Discard the used pipette tip. To avoid contaminating the matrix, always use a new tip for every spot. Step 3. Add a bacterial isolate or additional bacterial QC organism to a sample spot. Using a 1 microliter loop, pick up part of a suitable colony or colonies from an 18 to 72-hour patient isolate grown on a validated media. Colonies on agar plates may vary considerably with respect to size, texture, or viscosity, so some judgment is required to obtain the correct amount of sample. Avoid picking up agar along with the colony, as this may lead to poor identification results. Apply the isolate to the center of the first available sample spot. Depending on the amount of material selected, dabbing 2 to 3 times with the loop works well. Flip the loop and smear the bacterial isolate in a thin, even layer on the spot. Be careful not to spread the bacterial isolate outside the spot limits. Good smear quality is essential. During testing, the goal is to acquire 100 profiles. If less than 30 profiles are obtained, the spot fails and the next spot is tested. Discard the used loop in an appropriate biohazard container. Open the VITEK MS-CHCA matrix file. It is not necessary to warm the matrix to room temperature. Immediately after spreading inoculum, add 1 microliter of clear matrix to the center of the spot. Do not allow the sample to dry before adding the matrix. No more than 2 spots should be prepared before the matrix is added to ensure the sample is moist when the matrix is added. If isolates are to be tested singly, matrix should be added as each spot is prepared. If isolates are tested in duplicate, matrix can be added after both spots are prepared. Discard the used pipette tip. Always use a new tip every time the matrix vial is entered. Step 4. Add a yeast isolate or additional yeast QC organism to a sample spot. Using a 1 microliter loop, pick up part of a suitable colony from a 24 to 72-hour patient isolate grown on a validated media. Picking up agar along with the colony may lead to poor identification results. Apply the isolate to the center of the first available sample spot. Depending on the amount of material selected, dabbing 2 to 3 times with the loop works well. Flip the loop and smear the yeast isolate in a thin, even layer on the spot. Be careful not to spread outside the spot limits. Good smear quality is essential. During testing, the goal is to acquire 100 profiles. If less than 30 profiles are obtained, the spot fails and the next spot is tested. Discard the used loop in an appropriate biohazard container. Open the VITEK MSFA formic acid vial. It is not necessary to warm the formic acid to room temperature. Add 0.5 microliter of formic acid to the center of the spot. Discard the used pipette tip. Always use a new tip every time the formic acid vial is entered. Allow the formic acid to evaporate. This usually takes approximately 1 to 3 minutes. When dry, open the VITEK MSCHCA matrix vial. It is not necessary to warm the matrix to room temperature. Add 1 microliter of clear matrix to the center of the spot. Discard the used pipette tip. Always use a new tip every time the matrix vial is entered. Continue sample preparation for any additional bacterial or yeast isolates. If the prep station is configured for duplicate deposit, the second sample spot should be prepared adjacent to the first. If negative controls are required by your lab procedures, perform the following. For a bacterial negative control, add 1 microliter of VITEK MSCHCA matrix to an empty spot. For a yeast negative control, add 0.5 microliters of VITEK MSFA formic acid to an empty spot. Allow it to dry, and then add 1 microliter of VITEK MSCHCA matrix. Allow all matrix and/or organism suspensions to dry completely. This usually requires about 3 to 5 minutes. Verify crystal formation on all spots. This is visible and has a yellowish film. Store the slide at room temperature, protected from dust and aerosols, and test within 72 hours.